where he pioneered the study of stochastic gene expression. That is actually a different type of single cell measurement, primarily by fluorescence. In 2012, he moved to hubrecht and embarked on single cell genomic techniques and its application to developmental biology and molecular biology. I now give the stage to him.
Thank you very much, Sunny. Thank you very much for organizing this conference and inviting me. It's really weird times, and I'm very happy that I can still have some feeling that I'm going to a conference, because I'm really missing interacting with my colleagues, international colleagues. Especially would really like to be in Beijing right now with you, but that's not possible. Enjoying the good food another time. So what I want to talk about is unpublished work that we did, where we integrate multiple lineage measurements from the same single cell in a model for colon cancer evolution. So let me first take a step back, and you already mentioned it before. If you go about 10 years back in time, my lab was mostly doing microscopy, and we were still studying single cells. We were making movies and looking where the cells are with respect to each other. But the problem was at that time that we can only measure 1 or 2 genes at a time. We were limited by the number of colors of the fluorescent reporters. And there was this other field that we kind of were jealous about, which were the omics people. They were sequencing genomes and sequencing transcriptomes, but they needed millions of cells. So there was kind of, we were jealous, but we understood that it's not really, we couldn't really compare, because they had to combine millions of cells in a tube, and we could do single cells. And nowadays, we're really in the best of both worlds, where we can do many cells and at the same time also look at many genes. So that is really exciting times. But as you know, with this kind of combination of things, it's not always the best of both worlds, because there's always something that has to sacrifice, right? And one of the things that we are kind of interested in trying to to save during this convergence is time. Because of course, when you were doing microscopy, you could make beautiful videos, you had perfect control over time. But nowadays, if you dissociate the tissue, you don't really know what happened to the cell before you dissociate it. So there's loss of the temporal information. And of course, also the spatial information is lost, and our lab is very interested in that as well. But I want to talk today about how to reconstruct temporal information. And of course, we already heard beautiful examples yesterday and today as well, is how you can do it. For example, we just heard in the previous talk that you can use RNA velocity. You look at which genes are already spliced, which are not. So the not spliced genes are the most recent RNA, whereas the spliced genes are already have been generated some time ago. So you can, by comparing these two populations, you can estimate where cells are going in time. And the dynamics there is of a few cell cycles. It's not very long, it's about 1 or 2 cell cycles. We also heard yesterday from Rickard about RNA labeling, metabolic RNA labeling. Our lab is also very interested in that. This also allows you to look at old and new RNA and basically make an estimate of where cells are going. Again, this is of the order of a few cell cycles. If you want to go back in time more than a few cell cycles, you have to do lineage tracing. So you somehow have to label cells during their life and read out the labels at the moment that you do the experiment. It's very much interest in our lab, and then over the years we have been developing different strategies for this. This is one example that we call scar trace. Many other labs have very similar methods, and there you use Cas9 and a guide RNA, for example, that you inject into an embryo to scar the genome. And this will happen over a certain time, and if this is during the development of an organism, you can actually accumulate more and more scars during the development, and then at the end of the experiment you sequence the cells and you can infer what happened during the history of the life of the cell. Now this is very powerful, in particular in model organisms, but what I want to talk about you today is how to do it in humans. And we already saw, again, a beautiful talk by Yanni earlier on, for example, chromosome losses in lymphocytes. He also mentioned it's not clonal, so it might be difficult to use this for lineage tracing, but for sure, copy number variation in single cells is a good way to look at history. I'll show you some examples of this. Because of course we cannot use scar trace in a human setting. That's clear. You cannot use Cas9 to scar a human genome. So we have to think about other ways to infer history from human cells. Copy number variation I already mentioned. The second one is somatic mutations. Every time a cell goes through a cell cycle and duplicates the genome, a handful of mutations will be created that will, in a sense, record the history of the cell. Of course, the challenge there is that it's quite rare and very difficult to find these mutations in the huge genome. Beautiful talk yesterday by Fuchu that I basically copied one of his figures that you also saw yesterday, where he was studying the evolution of cells in tumors, in particular colon tumors. And typically what people do is they use these kind of illustrations to show the history of what happened during the life of cells. And for example, here there was an initial founder cell that grew into a large population, and then there was one cell here that created a new lineage by, for example, losing a part of chromosome 17, and then that creates a new clone, and within that clone more and more subclones arise. So this is how you can do inference of history in vivo situation, and beautifully done here in colon cancer system. The challenge, of course, always is that we only typically can do the measurement at the end. And ideally what you want to do is you want a time series and really measure during the phenomena that you want to study. This is very difficult. Here is again a sketch of how it works in colon cancer. So you typically start with a stem cell. That stem cell is healthy, has a healthy genome, and then over time this stem cell accumulates mutations, and these are called driver mutations. APC is one of them, is typically the first one, and then there are 3 other ones that will follow, turning that stem cell into a aggressive growing cell that will create a larger population that is now competing with the healthy cells. And over time, these cells accumulate more and more mutations and copy number losses, creating a very diverse tumor. And then at the end of this, the life of this tumor, the patient is already in the clinic, and of course you didn't have any data before that. So in an ideal experiment, we really want to kind of sample this process over time with periodic intervals. And this is, of course, at the moment very difficult to do in the clinic. So what I'm going to show you today is a model for this, which is based on organoids. And in organoids, of course, we have the freedom to grow them and let them evolve and sample cells whenever we want. So that's what I'm going to show you today, a model based on organoids. Now how this works is not developed by us, but our neighbor, Hans Clevers, developed this system where you basically start growing organoids from wild-type, healthy human colon. And then with CRISPR, you introduce the 4 driver mutation that I mentioned before, APC, P53, KRAS, and SMAD4. You basically introduce them by hand, and now you have a very clean system that has all the 4 driver mutations that make this system very aggressive. And then you can start to do experiments with this in the lab. So what we did is, in addition to these mutations, we also introduced a viral barcode in each cell. So this is just a complex nucleotide of 20 base pairs that is basically random, and then we introduce this in single cells so we can label each cell uniquely, and later when we sequence these cells, we can also find what are the family members of that cell, because they should share this viral barcode. So this viral barcode is introduced at the beginning of our experiment. So how we do this experiment is we take these lentivirally coded organoids, and then we do a very long-term evolution experiment. We first split this founder population in 3, so we do 3 replicates, and then we basically evolve this population over the time of about 25 weeks, which is about 100 generations of cells, so cell generations. And then, of course, since this is a system is very accessible to us, we can periodically sample the culture, and we do this typically every week. We sample the cells and we associate them, do single cell sequencing and also bulk sequencing. And for now, I will just mostly focus on the genome sequencing here, not so much the RNA, as you will see. Right, so this experiment is nice. We'll generate a lot of data, and I'm gonna first tell you who actually did this. So Leonard and Buys are two great students in the lab that really did the heavy lifting here. Leonard did really the majority of all the experiments, and Buys did all the analysis, which I will show you is quite challenging. And this is done in collaboration with Jeroen de Ridder, who is at the hospital in Utrecht. Okay, so the goal that we have is that we want to look at these cells, but we want to measure as many lineage marks as we can from a single cell. And we're going to focus on three different measurements. So one is the copy number change. I already mentioned it to you sometimes, and we saw it before this morning and this afternoon, that sometimes you lose a chromosome, sometimes you gain a chromosome. So this is, of course, a heritable change, and you can use this also for lineage information. So we will measure the copy number changes. More difficult is to measure the somatic mutations. So we will also do our best to pick up mutations that happened during the experiment of the evolution. And then also, as I mentioned before, we have lentiviral labels that we introduce at the start of the experiment. It's a very complex library. Basically every cell is uniquely labeled, and then we can measure those labels during the experiment, the frequency of those labels during the experiment. So let me show you all those three measurements separately first, and then I'm going to combine them all. And that will be the challenging part. And if you have any good ideas, let me know, because we're very much struggling with this, and we have some ways to do it, but it's a challenge. Okay, so copy number changes. How you do this? So I'm basically showing here now about 1,600 single cells, where I'm showing the whole genome from left to right, so chromosome 1, 2, 3, etc. And what we're doing actually here, I won't go through the details, but we use a restriction enzyme approach. So we basically digest the single genome, and we ligate adapters to the sticky ends, and then we use in vitro transcription, very much like Lianti, actually, to amplify this. And what you can see here is that most of the cells are diploid. So when you see gray here, it means it's a diploid cell. But sometimes there's little blue parts here, which means you actually lost a little piece of the chromosome, in this case 1. Now you see that actually all the cells that I'm showing you here lost this. So this is actually a aberration in chromosome 1, which is actually in the founder cell. So this is kind of positive control. You want to see this in all cells. But more interestingly, chromosome 4, for example, sometimes you lose the entire chromosome 4, one copy of it, but not all cells. So these cells still have it, but there's few cells that really lost it, and so they form a clone here. Now there's also gains, which are the red signals here, and you can beautifully see that there's subclusters of populations that share copy number variants. So this is our first measurement to measure basically this lineage. Now just zoom in on one of those copy number changes, you can really cleanly identify it, and I think with typical resolution that we get is about 100 kb here. Of course, it depends very much on how sequence. Now what's interesting is that these copy number changes are not random. In this model for colon cancer in these organoids, when you compare the copy number losses to real tumors that are from the clinic have been characterized for the karyotype, you find very similar changes in the chromosome. So for example, chromosome 4, I mentioned before, is often also lost in real tumors, and the gain of chromosome 13 is also clearly visible in the organoids. It's also something you see in the clinic. So it's very interesting that these organoid models seem to mimic really the gains and losses of chromosomes in real colorectal cancer samples. All right, so let's go to the somatic mutation. So this is way more difficult to do, but it's possible. So what we basically do is, what's really important is that you need to phase those mutations, those somatic mutations, to SNPs, to basically what I'm calling here the genome SNV. So these are what we sometimes call SNPs, right? So these are basically in every single cell that there will be there a nucleotide that's not referenced. So we use it as an anchor, and then we search around those anchors for somatic mutations. And this has been recently published to two independent labs how you can do this well. And this allows us to really, there are a couple of advantages. One is that the noise really goes down, because of course you can really see a lot of mutations if you just take the raw data. These can be sequencing errors or mapping artifacts. But if you phase them with these SNPs, it's actually, you get a much higher significance. The other thing is you can both measure the presence and the absence of this particular mutation. And that's what you see in this matrix. So blue here means that this mutation is basically absent, so it's reference. So it maps to the reference of the genome. And red is where you actually have a mutation. If there are about 300 cells, you can already, for the raw data, see that there are basically 2 clusters of cells here that have 2 sets of mutations. This is additional information. So measure next to the copy number variant. And then the third measurement we can do is, of course, we can measure those lentiviral codes in single cells and realize, of course, that these are introduced only once at the beginning of the experiment. And during the experiment, they don't change. We don't reintroduce them. So if you see any difference in the frequency of these labels, it means that there is selection going on for certain cells. And that's what you, for example, can see here. So these are three replicas. Here left is the beginning of the experiment, and here is the end of the experiment. You can see indeed there's about 100 generations. You start with almost 100,000 different clones at the beginning. You cannot see all the colors here. But what's quite interesting is that over time you see that there is actually quite a bit of selection going on. And at the end of the experiment, you're only left with about 5 to 15 different clones, whereas we started with 100,000 cells at the beginning. So this way we can actually look for selection of the different genotypes during the experiment. So this information also we have at the single cell level. This, of course, at the population level, but also at single cells, we have the lentiviral barcode also identified. Okay, so now what's really a challenge, and this is what we spend most of our time on actually, is try to really combine all these 3 measurements together in the same cell. It's a challenge, not only conceptually, but also to present it. So I'm going to do my best. But I feel it's still not perfect. So I'm going to show you some examples. So this is a tree that we obtain just based on the copy number information alone. So now I'm ignoring the lentiviral information and the somatic mutations. So here on the x-axis you have time in weeks, and this is the founder cell that then grows out. And again, every week we have data, so we can basically look at the fraction of all the different copy number variants. And the width of these bands here, of these trees, is how many cells relatively there are. So if you see a very broad kind of band here, it means that lots of cells are in this particular copy number clone. Some copy number clones die off during the experiment. But what you can see is that there's initially a very large clone here, which is actually the loss of chromosome 18. So all these cells here, they lost chromosome 18. And then somewhere halfway in the experiment, there is another branch starting here where, in addition to chromosome 18, also chromosome 4 is lost. Turns out we can, since we have both bulk data and a lot of single cell data, we can actually phase all the SNPs on the chromosomes and we can get allelic information. So this is the loss of one of the alleles of chromosome 4, say the paternal allele, and this will be the loss of the maternal allele. So you see that happens both independently. So this, again, this tree is only based on the copy number. And we have about 30 different copy number states, but in this particular experiment, at the end of the experiment, only 3 or 4 of these copy number states survived. And now I'm going to project on this tree the lentiviral information. They will look like this. So now colors now indicate the lentiviral barcodes, and again the width of these colored bands mean how much what is the relative amount of lentiviral barcodes at that point in time. So let me give you an example. So when you, for example, here at 10 weeks, we have this big clone which lost chromosome 18, and there are at least 5 or 6 different lentiviral barcodes in that clone, right? So many different clones here that are identified by the lentiviral barcodes all have lost chromosome 18. Now this was something we actually did not expect, because we expected that a particular copy number clone is seeded by a single cell, and that single cell should be clonal. So you would actually expect only one lentiviral barcode in here. So, of course, there are other explanations. So it could be that the heterogeneity was already present at the beginning of the experiment. So when we started this experiment, and we labeled the cells with the lentiviral barcode, perhaps there was already a subpopulation that lost chromosome 18. And that's why there are multiple clones now that lost 18 and have different lentiviral barcodes. But if you look at the copy number profile of the ancestor, and the chromosome 18 is here, there is at least no strong evidence for that. Of course, it might be a very small population that we didn't see. So, but we should keep this in mind. We cannot rule this out 100%, but it's, I would say, unlikely, because otherwise I would have seen like some hint of a loss of chromosome 18 in the bulk data, but we don't see it. The second hypothesis is that these copy numbers are not rare. These changes in the copy numbers are not rare, but they happen all the time during this evolution experiment, independently in different cells. So it's not a rare event, but this happens all over, many, many times, recurrently. So keep that in mind. I will come back to this to test this hypothesis later. But what you can also see already here at the end point, so when you now look at this clone that lost chromosome 18 and 4, there's also 2 lentiviral barcodes in there. So again, this could either be that it's at the beginning of the experiment, it was already the cell that lost both. But again, this becomes very unlikely because now these are multiple losses of chromosomes. The other thing which is important to realize is that we can identify the loss of the paternal and the maternal chromosome independently. So you see that chromosome 4, maternal and paternal, are both lost. So at least that is for sure something that happens independently in 2 cells. Now, the third experiment I'm going to show you is the one where we now combine the copy numbered clones with the mutations. I'm showing it basically in a similar way as for the lentiviral barcode, but now there's only 2 colors because either the mutation is there or it's not there. So when it's green, it means that the mutation is referenced, so there's no mutation. When you see a red square, it means that the mutation is actually in that cell detected. And what you see here, and this is again the same experiment, is now that most likely these mutations, so this is really one single nucleotide somewhere in the genome, these mutations happened during the experiment because it was not, we didn't see it before, but then we see it, and then there is a polyclonal behavior here in this chromosome 18 and chromosome 4 loss, because they lost 2 chromosomes, but also they have both the mutation present and absent. Now, this mutation is not a driver mutation. We see it somewhere in a non-coding part of the genome. So we can use this as a kind of a lentiviral barcode that was introduced during the experiment. It's very unlikely that there were already 2 subpopulations at the beginning of the experiment that both had the somatic mutation and lost 2 chromosomes. So it becomes now very unlikely that this happened at the beginning of the experiment. So what we think is happening is that you can see it for both alleles of chromosome 4, that these events are really recurrent and happen over and over again. And these are independent losses of chromosome 18 and 4. And also the chromosome 4 is lost independently over time. And again, this is just one example of one particular somatic variant in the genome, but you can find many, many more. I won't go through all of them, but this is an example where I show you 3 other ones. So these are chromosome 13, 4, and 9, and they have exactly the same behavior. So this is something that is not based on one particular mutation in the genome, but this is pretty common, and we see this all over the time, all over the place. And of course, these chromosomes are diploid; they're not gained or lost. So these are at the copy number 2. So this is really these are really valid mutations that we can use for lineage tracing. Okay, so let me go back to this sketch that I had before, right? So basically, how we think about the generation of heterogeneity in a tumor is that there's a founder, but at a certain point, the tumor just becomes more and more heterogeneous because there's independent events in all these cells that generate different genotypes, different mutations, different copy number losses. So all these shades of color, typically at the end of the experiment with a dashed line, each of these clones you consider to be a different tumor clone. That's, I guess, how most people think about this. I think what we see now is that we have to be a little bit careful, at least for copy number variants, I think. Might not be true for somatic mutations, but for copy number variants, I think a model like this is more likely that actually if a single color red here means that you lose the same chromosome or gain the same chromosome, this happens over and over again, I think, during the evolution of the tumor. And although they might still be different because there might be other things that are different other than the copy numbers, in terms of the copy number profile, they're very, very similar. And what we think is happening is these copy number changes occur recurrently and with pretty high probability. Now, I already showed you that these organoids are not randomly losing these chromosomes. They basically lose the same chromosomes as you would see in the clinic. So we also looked for this particular case of chromosome 18 loss followed by chromosome 4 loss, and this is also happening in the clinic. So what you see here is basically a two-dimensional histogram showing the ratio of chromosome 18 on the x-axis and 4 on the y-axis. So you see that a lot of patients are here where they lose both chromosomes. And from this data, you can also kind of infer the sequence, because it seems that you first have to lose chromosome 18 and then you lose 4. It's not the other way around, that you first lose 4, because then there's no statistics here. So it suggests, the clinical data suggests as well that there is a sequential loss of these two chromosomes also in clinical data. Showing you this to give you some justification why these organoid models are a reflection of what's happening in the clinic. Now, when you look at the Kaplan-Meier curves for these particular combination of chromosomes, it's also a chromosome combination that leads to a higher mortality. So here you would have only chromosome 18, and this is chromosome 18 with 4. And if you go through the different statistics, it's really this combination that really makes a difference. Okay, so I'm at the end of the talk. The conclusions: we have developed a single-cell sequencing method to measure both copy number changes, viral barcodes, and somatic mutations in a single cell. And then we can compare all these three measurements to see if we can make consistent conclusions about what's happening. The main take-home message I want to give to you is that it seems that these copy number changes, they happen a lot. They're, of course, also strongly selected for. But I think we have evidence that this is an event that happens many times in the same population of tumor cells and leads to basically the same copy number profiles. But these are not independent events. They happen in many cells, independent cells in the population. And I also have to I try to show you that the things we observe are not random events that are standing by themselves, but they are very much reflect observations in real tumors, hopefully showing that this organoid model has some relevance. Now, I want to thank, again, Leonard and Buys very much for working so hard on this, but also our entire lab and the funding sources. Very enthusiastic group of people. I'm very happy to work with them. And thank you for your attention.
Thank you, Alexander. Very interesting talk. Can I ask you the first question? If I understand correctly, I want to confirm this is right. You were there at Yan Yi's talk, right? At the end, I asked him why the copy number variation of chromosome 21 is always there, and his answer was, the basically the apoptosis of those cells. It's likely all this copy number variation occur with equal probability everywhere, but when the copy number changes, the gene expression, the dosage, the protein dosage are different, and the cells have different probability of survive. And the 21st was one that it just survived. And is that what's happening? Because I see the cell cycles you went through, up to like a 25 or something, or there's something else. I didn't quite understand the I didn't quite understand the viral. What do you talk about later on? Is this something just the survival of the cell and that give you the preference for certain copy number variation patterns? Those are simply the ones that would survive the.
I think, of course, in a cancer background, it's more complicated, right? So the Yanji's data was basically in healthy cells. So typically, losing a chromosome or gaining a chromosome is lethal, indeed, and perhaps chromosome 21 is an exception. I guess in cancer background, it's much more tolerated because you have these driver mutations that basically switch some of the control mechanisms off. But then still, I think most of the combinations are lethal, even for cancer cells. But there are a few of them, and I guess those are the ones that we're picking up that are not lethal and actually give a growth advantage to these cells. Like chromosome 18, chromosome 4 seem to be actually enhancing the fitness of these cells. And because there's the control mechanism has been mutated, I think this is tolerated. And these lentiviral barcodes, yeah, we basically use them to measure the relative frequency of the different clones over time. That allows us to basically, if we now link that lentiviral barcode to the copy number, we say, okay, that copy number clone has this frequency. Yeah.
Okay. Yeah, thank you.
Hi, Professor. I have a question about the organoid. I want to know the organoid was developed from many cells or from the single cells. As you mentioned that you found a copy number variation, maybe some rare cells, they have copy number in some chromosome you didn't detect it in the bulk samples.
Yeah. Yeah, that's a good point. So of course, these lines were all made clonally, but of course, I think one take-home message that we also learned from this is that you can start with a clonal population, like of course with a single cell, but even if you grow them up, they will already start to diversify. I think these organoids are probably never clonal, I think, if you really think about it, because they are so dynamic. And we also did these experiments with wild-type organoids, and there the dynamics is less, but you still see there's quite a bit of selection. So yes, we start with a single cell, but the single cell has to be grown up to a certain amount so that we can actually sample from it, right? So there will, at the moment that you seed the single cell, the diversity will start to be introduced already. But yeah, there's nothing we can do about it. But in a sense, that's also interesting because that's what we're studying. But there's I think no way to keep these lines really clonal.
Yeah, and another question that you observe that the mutations and the lentiviral barcodes based on the copy number variation trees. I want to know if you construct the tree based on the mutations or the barcodes, if the phenomenon will not as I mean that maybe the mutations and the lentiviral barcodes, they are more coincident compared to the CMV tree.
Yeah, so of course the lentiviral barcodes are introduced at the beginning of the experiment, right? So if you would make a tree, it would basically be horizontal lines because the clones are identified at the beginning of the experiment. We are thinking about how to make trees from the single cell somatic mutations. The challenge there is that it's still very difficult to get a lot of coverage of the same nucleotide across thousands of cells. So what we do is, as I said, we use this restriction enzyme to digest. And then what we do is we identify about, with a very deep sequencing experiment, we identify, about 500 to 1,000 candidate mutations. And then we do many more cells and we pull out those regions with a bait strategy to get higher coverage. So this helps, but still the dropouts are still a problem, so that the trees are not as resolved as the trees for the copy number variants, which are, we have much more statistics. So, but it's something we're working on to see if we can make two independent trees and somehow join them. This is a challenge. We don't have a solution yet.
Okay, thank you.
Alexander, it's a great talk. So maybe I missed it. So have you tried to compare the, how to say, when you, for example, transplant these unknown back to the new mice? So do you see the difference between these copy number gain or loss and the normal ones?
Yeah, no, that's a great suggestion. We tried some of the experiments. They didn't work out because of technical reasons, but this is a very interesting thing to do.
You mean no difference, or just you haven't tried?
No, we tried, but we ran into technical things, nothing to do with the hypothesis. We have been trying, but we haven't been successful. But this would require more time, to put them back into an organism.
Okay, thanks.
Hi, Alex. In your lentiviral labeling system, in the beginning, I just wondering, is there some preference for this barcode integrated into the host genome, and do they have the association with the later survival of the chromosome lossing or amplification?
Yeah, no, you could of course expect that one of these lentiviruses goes into like a tumor suppressor or something like that, right? So that so we didn't characterize the location of the lentivirus. Now, I guess what we do is we have multiple replicas, and all the conclusions that we have, they are basically only seen in all these replicas. So I think it's unlikely that it's a lentivirus that really affects the genome or the phenotype of the cell. But it's something, and of course we also have the somatic mutations and the copy number losses independently, and we also have a control experiment where we don't have the lentiviral integration. But we don't have measurements on where this lentivirus has been integrated. But I would say it's unlikely that this is the driving force, since we see this reproduced in multiple experiments, also in experiments where we don't have the lentivirus.
Thank you.
Alexander, the genesis of cancer, whether it comes from copy number variation at the very beginning or the point mutation, of course, the frequency of copy number variation is low, but when it happens, it affects many bases. What do you think for your organoids system?
Yeah, no, I think what we learned here is that I think that indeed it is a rare event to lose a whole chromosome or gain a whole chromosome. I think, but at the same time, there's so much selection going on that if you combine those, that it becomes actually the probability that this happens multiple times independently in the same tumor is actually reasonably high. That's, I think, the only way we can explain experiments. And I think this is something we have to think about, how that affects treatment of a real tumor. I don't really have any very strong ideas there, but I think we have been treating this as kind of a rare event that grows into one big thing. I think it's many of these events that grow into multiple clones that are very similar. I don't know what the implications would be for treatment, for example, but definitely looking for vulnerabilities around copy number gains or losses, I think would be something important to think about. But yeah, so this is really the beginning. It would be very interesting to use the system also in combination with drugs, right? You grow them, add a drug at a certain point, see which ones survive. Then actually, because we have all this lineage information measured before and after the drug, we can hopefully link it back in time and see what the population before already primed to basically survive the drug or not. Yeah.
All right. Thank you, Alexandra, for the very stimulating talk. Is Ido here?
Yeah, hi.
Hi, good to see you, Ido.
Great.
So,
thank you for the wonderful presentation.
Yeah,